Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will talk about MacConkey Agar. In every microbiology exam, 2 to 3 questions are asked about MacConkey Agar. So it's a very important topic. So let's start. MacConkey Agar is a culture media that is used for the growth of gram-negative bacteria and especially for Enterobacteriaceae family and Pseudomonas. So MacConkey Agar is a culture media. Culture media is used for the growth of bacteria. Culture media, MacConkey Agar is a culture media for gram-negative bacteria especially for Enterobacteriaceae family and Pseudomonas. Enterobacteriaceae family includes E.coli, Salmonella, Shigella, Proteus, Klebsiella. So MacConkey agar is used for the growth of gram-negative bacteria. Not all gram-negative bacteria can grow on MacConkey agar. For example, there are certain bacteria which are known as fastidious bacteria, having special growth requirements. And fastidious gram-negative bacteria includes Haemophilus influenzae and gonococcus, which is also known as Neisseria gonorrhoeae. Those fastidious bacteria cannot grow on MacConkey agar. So only gram-negative bacteria that are enteric bacteria which are present in the intestine of humans and animals, such as bacteria belonging to family Enterobacteriaceae and Pseudomonas, can only grow on MacConkey agar. Also keep this point in mind. This is most commonly used media because it has two main properties. First, it is a selective media. And second property, it's a differential media. Media or agar, differential media or agar, it's the same thing. Selective media means a type of agar which allows one type of bacteria to grow but inhibit the other type of bacteria. So MacConkey agar is a selective in a way it only allows gram-negative bacteria, whereas gram-positive bacteria cannot grow on MacConkey agar. So gram-negative bacteria can grow whereas gram-positive bacteria cannot grow on it. So gram-positive bacteria cannot grow on MacConkey agar because MacConkey agar contains two substances. First is it contains crystal violet and second MacConkey agar contain bile salts. These two, crystal violet and bile salts, don't allow gram-positive bacteria to grow. So only gram-negative bacteria that are enteric, belonging to genus Enterobacteriaceae and Pseudomonas, can only grow on MacConkey agar. Second property of MacConkey agar, it's a differential media. Differential media means it is a type of agar which differentiate between different kind of bacteria on the basis of bacterial appearances, right? How bacterial colony looks like, this will tell us what kind of bacteria it is. So MacConkey agar is a differential media or differential agar. It differentiates between all those gram-negative bacteria that can ferment lactose, from those gram-negative bacteria that cannot ferment lactose. So MacConkey agar is a differential media, differentiate between lactose fermenter and non-lactose fermenter. You may see in certain books lactose fermenter are written as LF, right? Whereas non-lactose fermenter are written as NLF, non-lactose fermenters. So how MacConkey agar can differentiate between lactose fermenter and non-lactose fermenter? MacConkey agar contain lactose, which is a carbohydrate, a sugar. So all those bacteria that can ferment lactose and their colonies will be pink on MacConkey agar, pink to red, pink or red in color colonies of lactose fermenter bacteria. Non-lactose fermenter bacteria cannot ferment lactose, so their colonies will be colorless. Lactose fermenter bacteria includes E. coli, Klebsiella, Enterobacter. Whereas non-lactose fermenter bacteria includes Salmonella, Shigella, Proteus. Also, these E. coli, Klebsiella, Enterobacter, Salmonella, Shigella, Proteus, these belong to Enterobacteriaceae family. Whereas Pseudomonas do not belong to Enterobacteriaceae family, and Pseudomonas do not ferment any kind of sugar. It is also non-lactose fermenter. It can be differentiated on MacConkey agar. So Pseudomonas, when Pseudomonas grow on MacConkey agar, it produces mostly green color colonies. So in short, lactose fermenter produces pink to red color colonies and non-lactose fermenter produces colorless colonies. If we're talking about the composition of MacConkey agar, I will write over here, composition of MacConkey agar in this box. MacConkey agar contain lactose. Now we know that it also contains crystal violet bile salts, crystal violet CV, bile salts. MacConkey agar also contain a pH indicator which is neutral red. Neutral red is a pH indicator in MacConkey agar. When the bacteria that can ferment lactose, after fermentation of lactose, lactic acid is produced, right? So lactic acid will reduce the pH of agar, MacConkey agar, from 7.1 to less than 6.8, right? When pH is less than 6.8, this neutral red is convert this agar into red in color. So all those lactose fermenter colonies will be red in color, and agar surrounding those colonies will also be red or pink in color. Whereas non-lactose fermenter do not reduce the pH of agar because they have not broken down this lactose and their pH is not less. So this neutral red will be yellow in color or colorless or yellow in color. Other composition of MacConkey agar, it also contain peptone, also protease peptone, NaCl, water and solidifying agent, which is agar. So this is composition of MacConkey Agar. So if we summarize this MacConkey Agar, only gram-negative enteric bacteria can grow on MacConkey Agar. Fastidious bacteria such as Haemophilus and Neisseria cannot grow on MacConkey Agar. Gram-positive bacteria cannot grow on MacConkey Agar as well due to crystal violet and bile salts present in the MacConkey Agar. MacConkey Agar also differentiate between lactose fermenter that can ferment lactose from those gram-negative bacteria that are non-lactose fermenter. Example of gram-negative lactose fermenter bacteria are E.coli, Klebsiella, Enterobacteriaceae, and their colonies will be pink to red in color, whereas non-lactose fermenter colonies will be colorless. Examples of non-lactose fermenter are Salmonella, Shigella, Proteus, whereas Pseudomonas is also a non-lactose fermenter. Pseudomonas do not ferment any kind of sugar, so its colonies will be green in color due to its pigment. Pseudomonas produces other color colonies also. So if you have any question regarding this topic, you can ask in the comment section. Thank you.